Frederick Griffith and the Transforming Principle. Let's talk about Frederick Griffith. He was born in Eccleston, Lancashire, England in 1877, and he studied medicine at the University of Liverpool and then worked as an army doctor before finding passion in microbiological research. Griffith began working with bacterial pneumonia because of the 1900s flu epidemic, and he attempted to create a vaccine for Streptococcus pneumoniae, but did not succeed. Griffith then veered away from vaccine research and began studying two strains of Streptococcus pneumoniae, a benign R strain and a virulent S strain. These two strains of bacteria differed in physical structure. The benign strain had a rough exterior and defined edges, and the virulent strain had a smooth surface caused by an outer capsule that essentially allowed the bacteria to avoid being detected by immune cells, thus causing the virulent nature of the strain. With the knowledge he had on these two strains, Griffith proposed the question: is it possible for the genetic information that determines the virulence of a strain to be transferred from one bacterial cell to another? He believed that it was. In order to test his theory, Griffith created an experiment using mice. Griffith first injected mice with the S strain bacteria and observed that they were killed after the inoculation. Griffith then injected mice with the R strain bacteria and observed that these mice were left unaffected. Both of these were the expected results. Griffith then injected mice with a heat-killed version of the virulent bacteria. In this heat-killed strain, the cell itself was dead, but the cell components remained intact. With this observation and the use of another experiment, Griffith then discovered that if he injected the heat-killed S strain bacteria in the mice, along with the live non-virulent bacteria, the mice died. Not only that, but when the blood of the mice was tested, virulent bacteria was detected. This was a revolutionary discovery that proved the existence of a transforming principle. Griffith's experiments showed that genetic information from a dead cell with intact cell components, such as proteins and nucleic acids, could be transferred to a live cell. The nature of a cell could be changed through incorporation of new outside genetic information. This transformation was also observed to be heritable, as the subsequent generations could also inherit the virulent information. Griffith published his findings in 1928, and the experiment became famously known as Griffith's experiment. It was his work that led Oswald Avery, McLeod, and McCartney to discover that the transforming principle of the cell is DNA. Griffith died in 1941 at the age of 63, but his contribution